Well, hello, this is going to be the first in a series of videos about enzymes. Let's just jump right into it. So I'd like you to imagine for a second that you're talking to a colleague of yours, maybe they're an organic chemist, and you say to them, hey, I've got all of these different molecules that I'd like to put into, a waterish, sort of warmish, neutral pH environment, normal temperatures, and I'd like them to react and do an interesting coherent thing. And what your organic chemistry colleague would probably say is that's basically impossible, at least not in a typical, what they would call one pot reaction. And there's a couple of problems with this, but the two ones I'm going to highlight here are first, that the reactions are actually quite slow. Most of the molecules that I just showed there are stable at physiological pH under aqueous conditions, so it's just going to take a long time for them to do any kind of reaction. But even if we could make them react, say we increase the temperature or we altered the pH radically, we would end up with a complete and total mess, right? We would have no control over the outcome, which reactions happen versus which others. Of course, the molecules that I just showed in the cell do in fact all hang out in an aqueous environment. Those are all metabolites that are inside of your cells right now. And yet somehow we achieve ordered outcomes. We get specific reactions that are being done, and this is done using enzymes. And so what enzymes do fundamentally is they catalyze specific reactions under physiological conditions. So aqueous, low temperature, neutral pH. We're not having to add toluene or go to some ridiculous temperature or add a platinum catalyst or whatever you've learned in an organic chemistry class. Instead, we're just going to say this is going to happen in salty water at pH 7, just like the stuff that's in our cells. And the specificity of these is actually quite remarkable. So what I'm showing here is glucose 6-phosphate. This is an important metabolite in central carbon metabolism. When we get into our metabolism videos, you'll become very, very familiar with this if you stick around. Glucose 6-phosphate can be turned into this molecule called fructose 6-phosphate if you add the enzyme glucose 6-phosphate isomerase. But if you add a different enzyme, I don't know, glucose 6-phosphatase, you can turn it into just a glucose molecule with a free phosphate. If you add the enzyme phosphoglucomutase, you can actually move it from glucose 6-phosphate to glucose 1-phosphate, so we just move the position of the phosphate. And then if you add this glucose 6-phosphate dehydrogenase and NADP+, you can actually do an oxidation reaction where you change one of the oxidation states of this glucose molecule. And what's totally remarkable about this is that each of these enzymes catalyzes this reaction and no other, right? If you add glucose 6-phosphate isomerase, you're going to get out fructose 6-phosphate. You're not going to get out a free glucose. So there's exquisite specificity, even though they're all taking in the same basic substrate, you can get completely different outcomes just by your choice of enzymes. And so what I'd like to do in this video is do a quick overview of enzymes, make sure we're all on the same page about some of the words that we use in talking about them, their sort of basic architectures. We're going to talk about classes of enzymatic reactions, and then we're going to zoom in and talk about what we mean when we say an enzyme is specific. So let's get into that. So first of all, most enzymes are proteins, a few are RNA. They're all macromolecules, though, that are encoded by some sequence of amino acids in the case of proteins or nucleic acids in the case of RNA. They take on a particular structure, and that structure allows them to do the chemistry. And the common architecture that they take is they have an active site. This is a position that actually does the interesting chemistry. So the molecule that's being chemistry is being done on will bind at that site, and that's where the interesting things will happen. And then there's always a big chunk of the rest of the protein around it, and a scaffold that basically stabilizes that active site. What we'll see as we get into enzyme mechanisms is that enzyme active sites have weird things going on. They've got unfulfilled hydrogen bonds. Maybe they've got buried charges. All the sorts of things that you might think are not what you want to do to have a protein that folds and goes to a particular structure. So all the rest of this scaffold is basically to sit around that enzyme active site to let it be sitting there quivering, ready to do whatever cool chemistry that it does. When we talk about the molecules that enzymes work on, we call them substrates. So instead of reactants, as you might have normally encountered inside of a chemistry class. What we can say about this enzyme here, this enzyme takes as a substrate glucose 6-phosphate and turns it into fructose 6-phosphate. Now, interestingly, actually both of these are substrates, right? Because this enzyme will catalyze that reaction, but if you put this at a high enough concentration, it will actually catalyze this reaction. It go backwards and forwards. So depending on the context, we can call it either of these substrates. They're both substrates for which the enzyme can catalyze a reaction. I just hinted at it in the last one, but enzymes increase the rates of reactions, but don't change their directions. And so here with this reaction, and specifically, this reaction has a delta G naught prime of about 0, so a standard state free energy of close to 0, which basically means there's no preference. It doesn't really prefer going one way or the other. What that means is if we make a huge amount of glucose 6-phosphate, this reaction, the enzyme will make the reaction flow from reactants to products. But by contrast, if we make a massive amount of fructose 6-phosphate, this enzyme will just as happily make a bunch of reactants go to products, right? So it's not changing the favored direction of a reaction. It's only changing the direction that the reaction flows. Basically, it does not change the direction it flows. It changes the rate that it flows towards where it's going to flow anyway. So if it's going to flow from products to reactants, it'll make it do so faster. If it's going to flow from reactants to products, it'll make it do that faster. Another important point about enzymatic reactions is that these reactions are so much faster than their uncatalyzed rates for most biomolecular interactions that we can basically view a cell that has an enzyme, has the reaction turned on, and sure, there might be some quantitative tweaks to how on it is. It can be sort of tuned up or tuned down, but to first approximation, it's on if the enzyme is present, whereas if the cell doesn't have that enzyme, that reaction is just off. So a good example of this is this enzyme here, glucose 6-phosphatase, which I mentioned a couple of slides ago. This enzyme is important for the metabolic pathway hexokinase, gluconeogenesis. And gluconeogenesis is actually a pathway that's basically only run in a few tissue types. So liver cells, for example, run gluconeogenesis. They have this enzyme. Muscle cells don't, which means that basically this reaction happens in liver cells, but it does not happen in muscle cells, just because of which enzymes are being made. Last couple things about naming. Most enzymes have a name that ends in ACE that generally says something about how it works. So I just threw up 7 different enzymes here. So adenylate kinase works on ATP type and derivative molecules, the adenylate, the adenosine, that makes sense. You'll see in a second, a kinase is a particular type of enzyme that does a particular phosphotransferase. So you're like, that one makes sense. Sometimes they make no sense at all. So trypsin doesn't say ACE, doesn't say what it does. Turns out it's a protease that cuts proteins, but it's called trypsin for historical reasons. You also have ones like this one down here, the ABC type D-galactose transporter is actually an enzyme. Doesn't say ACE anywhere in it at all, but it's actually using ATP to move things across the bilayer. Just so you put this up here, I put this up here not because it's a pretty slide or I expect that everyone's going to learn it, but in the past, sort of a pre-Google era, like when I was taking biochemistry, you learned all these. You're like, you got to memorize all these enzyme types because it was, you really need to have them available while you're looking at things. Now, since you can instantly look up what a kinase is, maybe it's less important to have it in your mind. But I put this up here because this really is about the top 10 different types of enzymes you'll encounter. And my anticipation is as you continue to study biochemistry, the 11th time you look up the word kinase on Google, it'll sink in that, oh, this is always an enzyme that transfers a phosphate group, usually from ATP. A protease is always something that hydrolyzes or breaks peptide bonds in proteins. So as you start looking at enzyme names, you'll start at enzymes in biological context, you'll start encountering these names. They really do have some meaning attached to them. I'm not going to go through all of them. I'm putting it up here so you can, screen cap this and put this on your wall and, think deep thoughts about it or whatever. But just so you know, there is a sort of a logic to the way that enzymes are often named. Okay. That was our enzyme overview. Maybe a lot of that was review, maybe not, but hopefully now you've got a broad picture. We've got a broad language to talk about enzymes. Now let's talk about the classes of enzymatic reactions that can be done. And it turns out that life actually, I'm going to keep disappearing. Life actually doesn't do very many overall reactions, right? There's basically 7 different reaction types that can be done by by cells, by living tissues, living things. There we go. That's what I'm looking for. And the reason there's so few is actually because there's so few molecules, right? We basically have chenops, so limited number of atom types that are available, limited number of bond types. So if you look across the entire breadth of all of chemistry that can be done, organisms tend to do a fairly small list. And it turns out you can actually organize all of enzyme chemistry in terms of these seven high-level categories, and then break down all the rest of them in terms of sort of specific reactants and products. So I'll say very briefly what these do. So oxidoreductases do redox chemistry. Transferases, whoa, we're moving things around. Transferases move groups one to another. So basically, you take a molecule, a chunk of atom off of one, and you stick it onto another molecule. Hydrolases do hydrolysis reactions. They cut bonds. Lyases stick things together without together. Wow, stick, can't write and talk at the same time. Stick together without ATP. Isomerases rearrange molecules. So basically the stoichiometry, or the sort of number of carbons, hydrogens, oxygens doesn't change. They're just arranged in a different way. Ligases stick together molecules with ATP, so they require energy to do so. And translocases move across bilayers. And so the idea here is there's really not that much stuff that's happening when you look at a giant cell diagram. There's only this small subset of reactions. But of course, enzymes are specific, right? They do specific sorts of reactions on specific reagents. So of course, it can't stay this simple. One of my favorite ways to explore this is actually to use this thing called the Brenda EC Explorer. This is the Enzyme Commission Explorer. And what we'll see is each of these things can be broken down into subclasses. Annoyingly, this is the best URL that I could find that resolves to the thing I want to. So sorry for the terrible URL, but if you go to that URL, which I'll also put in the description, you can actually go and browse what I'm about to show you. So what I've done is I've just taken the thing from the top. This is the EC tree with its 7 different classes. And in this browser, you can actually click on each of them and expand them out. So here I've expanded out the oxidoreductases into this big block of different sorts of reactions that are catalyzed, and what you can see is they're categorized by the sort of chemistry they do. So these are all redox reactions, but here these are redox reactions that are acting on CHOH groups. These are redox reactions at 1.6 that are acting on NADH or NADPH, etc. So you can go through there and say, oh, what sorts of reactants do I have? You can then go to the next tier. So I just clicked on 1.1. Now I'm here, 1.1, and then look at 1.11. This is now, they specifically has NAD plus or NADPH plus as an acceptor. This down here, 1.1.9, has using a copper protein as an acceptor. So we're starting to get more granularity into the sort of redox reaction that's being catalyzed. And then we can open up that to the next level. So here, 1.1.1 is specifically alcohol dehydrogenase. So now we're down to an individual named enzyme has the number 1.1.1.1. This enzyme down here, 1.1.1.10, is L-xylulose reductase, right? So when we're looking at metabolic pathways, we're looking at how cells work and how they behave. What we'll see is we can often identify, or we can always identify a specific enzyme by its EC number. And so let's look at this one, alcohol dehydrogenase, 1.1.1.1. This is actually the enzyme that does the first step in the metabolism of ethanol. So if you drink alcohol, wine, beer, whatever, this is the first step in breaking that down. I think it's very odd and funny that the Enzyme Commission's like the first enzyme you find in all of their tables, 1.1.1.1, is this particular enzyme. But make of it what you will. Okay. The nice thing about EC numbers is they unambiguously identify enzymes by their chemistry and substrates, right? Remember we mentioned trypsin. Well, what the heck does trypsin do? We can actually look up and identify every enzyme with these numbers. So anytime you see an enzyme in Wikipedia or in a textbook, it'll be associated with like this number here, 3.4.21.4. If you look that up, that'll tell you, oh, this is an enzyme that cuts peptide bonds, for example. Okay, so enzyme overview, classes of reactions. Let's do our last bit on enzyme specificity, and then we're going to be done. Remember I said at the very beginning of this that enzymes are specific in the chemistry that they do. And or they say they're specific. Let's break it into two parts. So first, they're specific in the chemistry that they do. And what I mean by this is, look, we can look at these EC numbers. We can say, oh, this is a 5. That means that this enzyme does isomerization. And indeed, what we see is it's just rearranging where groups are on the same molecule. We look up here at this one, and we say, oh, it's a 1. That's an enzyme that's involved in redox. We look out here at this one, and we say, oh, this one's involved in hydrolysis. And this one over here is involved in isomerization. And the basic idea is that because of how these active sites on enzymes are arranged, basically an enzyme that does redox chemistry is not one that can do isomerization, or an enzyme that does hydrolysis is one that can't do redox, right? So enzymes are specific in the sorts of chemistry that they can catalyze. So that's one level of specificity. But the second specificity is they're specific in the substrate that they recognize. So for example, here's glucose 6-phosphate isomerase. It catalyzes this particular reaction using this particular sugars. But a closely related sugar is one that it just won't be able to recognize, right? So it can take glucose 6-phosphate and make glucose 1-phosphate. But if you give it a closely related sugar like galactose 6-phosphate, it just can't recognize it at all. So they're specific in what substrates that they recognize. And these substrate recognition rules, recognition actually follows the rules we've talked about in previous videos, talking about non-covalent specificity. So when I was talking about intermolecular interactions, I talked about how we can break non-covalent interactions into nonpolar and polar, and said that the non-polar interactions like the hydrophobic effect tend to just make things stick together, whereas things like hydrogen bonds and ion pairs tend to basically make only specific molecules stick together. And so when we're asking a question like what substrate is recognized by which enzyme and how and why, we're going to be thinking about it in exactly the same way that we've talked about in the past, thinking about things like, well, how does the estrogen receptor recognize estradiol versus testosterone? We see the same rules applying when we're talking about enzymes. I thought I'd go through this with a specific example, actually, really quickly, and that is the example of protease enzymes. So this is one of them called trypsin. And what trypsin does is it actually cuts peptide bonds right here that immediately happen after a lysine or arginine in the primary sequence of a protein. So this is actually something that exists in our digestive tract. It's basically helping us break down food, and it's doing so by cutting. You eat a tofu burger. You want to break down the protein in tofu into its amino acids. This is one of the enzymes that's involved. Enzymes that is involved. So I'm showing you the structure of the protein here with the substrate docked onto it. I'm going to pull them apart and we're going to look at them. So I'm going to pull just the substrate out, and then I'm going to turn the substrate 90 degrees. And what hopefully you can see on this structure is that there are different regions of the proteins that have different colors. Those colors are what's known as an electrostatic map. So basically you look across the protein structure, take an average of whether the amino acids tend to be more positive or more negative at those positions and sort of smear it across the structure. And what I'd like to point out right here is this really, really deep pocket. There's a deep hole, and that deep hole is negative at the bottom. It's red. That implies that there's a deep negative pocket in that position. If you look at, I'm going to rotate it again. So this is the substrate. I'm going to rotate it 90 degrees. That's what this other angle is. And then I'm going to put the same surface on the substrate. And hopefully what you can see is there's a really blue positive part. That's the lysine. And so this molecule, trypsin, can recognize peptides that have lysine in them because lysine, that positively charged bit, sticks into that negative pocket, and it gives it specificity. So I'm showing that here schematically. I'm going to make myself disappear again. So trypsin has a deep negative pocket, and it binds to a positively charged lysine. Let me show you two other proteases in the same orientation. Chymotrypsin, which has a deep hydrophobic pocket, and a molecule called granzyme, which has a shallow positive pocket. And just this is a good exercise really quickly. Think about it for a second. If you had to guess which enzyme, these both cut after a particular amino acids, what's the preference? What does alpha chymotrypsin do? What does granzyme do? The options are an aspartate or a phenylalanine. So maybe pause and think about it. All right, hopefully you've thought about it. Deep hydrophobic pocket, that's going to recognize things like phenylalanine and not things like aspartate. whereas granzyme is shallow and positive and it has a very strong preference for aspartate over other things. And so by changing the aspects of the chemistry in the same way that, say, we saw changing the aspects of the chemistry in the steroid receptors or other places, enzymes can bind to specific substrates to apply their particular chemistry. Okay, so what have we said in this video? We said that enzymes catalyze specific reactions under physiological conditions. The presence or absence of an enzyme can really determine whether a reaction is on or off in a cell. So reactions are sped up so much by enzymes that if an enzyme's around, it's on. If an enzyme's not around, it's off. And then we talked about how enzymes recognize specific substrates and enable specific classes of chemistry, allowing them to overcome this problem. If you just put a bunch of small molecules into an aqueous solution, how in the world do you get specific outcomes? Okay, that's what I have about enzymes. Hope you enjoyed it. And we've got another stack of videos coming out. We're going to talk about things like how enzymes speed up reaction rates, how to measure reaction rates. So I look forward to doing that with you, and see you later.